Good day. My name is Roland Benedict M. Espinosa, and today I'll be presenting a study titled Activity Detection of Lactic Acid Bacteria based on Fluorescence Color Difference of Boron and Nitrogen Co-Dope Carbon Dots, published in Food Bioscience Journal in 2025. This research explores an innovative fluorescence microscopy technique for detecting bacterial viability using boron-nitrogen co-dope carbon dots, or BNCDs. A quick background: lactic acid bacteria, or lab, are widely used in food fermentation and probiotic production. Monitoring their viability is crucial for ensuring product quality and optimizing industrial processes. Traditional fluorescent dyes used for viability detection have major drawbacks: high cost, toxicity, and instability. This study aims to overcome these limitations by developing an alternative using BNCDs. The study focuses on these four main objectives. First, synthesize and characterize BNCDs as fluorescent markers. Second, investigate the fluorescence properties of BNCDs under different conditions. Third, evaluate BNCD's ability to differentiate live and dead lab cells based on fluorescence color. And lastly, assess BNCD's applicability in imaging other cell types beyond lab. To achieve these objectives, fluorescence microscopy was used to observe lab cells labeled with BNCDs. Fluorescence microscopy uses fluorescence and phosphorescence instead of reflection and absorption. The BNCDs were synthesized using citric acid, ethylene diamine, and phenylboronic acid via a solvothermal method. The preparation of lab cells were divided into three groups: live lab, dead lab, and mixed lab. The microscopy setting is done either with MLab with different BNCD concentration or with lab in different excitation wavelengths, namely UV at 350 nanometers, blue at 480 nanometers, and green at 530 nanometers. These are a few additional methods used: fluorescence spectrophotometry, UVB spectrophotometry, transmission electron microscopy or TEM, X-ray photoelectron spectroscopy or XPS, and FTIR spectrophotometry. It can be observed from Figure 3A that BNCDs exhibited concentration-dependent fluorescence properties. Higher concentration means higher fluorescence and also higher background noise. From the clarity and distinctness, the optimal concentration of BNCDs for labeling LAB was observed to be between 200 and 400 grams per milliliter. From Figure 3B, it can be observed that at different wavelength excitations, the different lab activities display distinct and different colors. Under UV light, L lab and D lab exhibited blue and green colors, respectively. Under blue light, both appeared green and yellow, while under green light, both were deep red and bright red, respectively. From Figure 7, it can be observed that the fluorescent microscopy imaging using BNCDs could label other cell types such as macrophages, onion cells, yeast cells, Escherichia coli, and Staphylococcus aureus. Fluorescence microscopy validated BNCDs as effective fluorescent markers, satisfying Objective 1. In Figure 3A, fluorescence microscopy confirmed BNCD's concentration-dependent fluorescence shift, satisfying Objective 2. In Figure 3B, fluorescence microscopy demonstrated that BNCDs could successfully differentiate live and dead lab with distinct colors, satisfying Objective 3. In Figure 7, fluorescence microscopy confirms BNCD's applicability to other cells, proving that this technique can be broadly used in biological imaging, satisfying Objective 4. This study presents several strengths. BNCDs are cost effective, easy to synthesize, and highly fluorescent, making them a viable alternative to expensive dyes. Another, they are non-toxic, ensuring safe application in live cell imaging. Another is that they demonstrated high fluorescence stability even under prolonged exposure. However, some limitations remain. The optimal BNCD concentration varies across microbial species, requiring further study. Large-scale industrial application also need validation before implementation. Overall, this study successfully demonstrated that BNCDs combined with fluorescence microscopy offer a novel and reliable method for detecting bacterial activity. This technique proves a cost-effective, non-toxic, and stable alternative to conventional fluorescent dyes, with broad applicability across different biological fields. Future research should focus on refining BNCD formulations and testing them in real-world industrial settings to maximize their impact. These are my references, and thank you for listening.